I'm perfectly aware that I'm the last thing that keeps you from lunch. And I'm quite terrified by it because recently I was reading in Kahneman's wonderful book, Thinking Fast and Slow. And in this book he describes a study that was published in PNS many years ago, where, hold on, the study reviews a committee in Israel reviewing parole cases over a whole day, I think over a whole week, and there are fixed days on when these people eat. And parole apparently dropped to 0 just before lunch, and so that's part of my concern right now. But Alain, 1.5 hours, is that right?
I'll keep a close eye on you.
I won't do it, don't worry. So, this is not quite working yet. Oops. Okay, so my topic is morphogens on the move, and in particular, evolution of morphogens. And when I started to become confronted with this concept a long time ago, I thought I knew what it means, but then a couple of years ago, I started to read a little bit about the historical origin of this concept and got very confused. And I actually think that it may mean quite different things to different people. So obviously, it has been brought up already. There is the theoretical approach that was started by Turing. And there's this, an experimental approach that started actually long before Volpert, but Volpert was very, of course, very much quoted for it. And I bring this up because I think these concepts have quite different implications for how we think about morphogens in evolution, and I wanted to summarize that briefly here. So obviously for Turing, morphogens are diffusing. There are at least 2, and they are interacting. And in evolution for him, he doesn't use the word evolution actually a single time, but it is quite clear that he has organismal diversity in his mind, and it is about, to achieve organismal diversity, it is about changing morphogen dynamics, so morphogens themselves driving evolutionary change. And for Volpert, who had a much more static concept, at least in his early papers, and was concerned actually with quite different questions, namely the scale invariance of gradients in fields of cells. For him, obviously, the dynamics of morphogen gradients were not the source for evolutionary diversity. Again, he doesn't use the word evolution, but in a later paper, he makes that case, actually. And I take these 2 quotes. The one is just the very beginning from Turing's abstract. And the second makes clear what Volpert was thinking about morphogens in evolution, a completely passive role. Where the responding genes are the stuff that makes evolutionary change. Now, I have been interested in both sides. I have been interested in the evolution of the dynamics of morphogen gradients and about the evolution of actually the genes that encode the morphogens. And I will talk about the latter today. But I do want to mention that we have done work on the other stuff too, and just briefly give you a couple of references there. Now, I go back now to some of the early evidence that inspired gradient and morphogen thinking in the Volpertian tradition. And one of the most beautiful examples I can think of are these double abdomens here shown for a chironomid midge. That's a picture from Kaltov's work, but actually the first who discovered this was Hideo Yajima in Japan, and he he also found that it is a phenotype that you can easily induce by exposing embryos to UV light, and later Kalthoff localized this more precisely and found that the anterior cortex is very sensitive to it. And that led, among other evidence, these earlier researchers to the idea that eggs have localized RNA, and that this might be the source of a gradient that has this long-range patterning effect. So obviously the first gene of this kind that was discovered to prevent this kind of phenotype was the bicoid gene in Drosophila. And what you see here is a tree of flies, and you see where bicoid has been found. That's these green little squares on the right. And you also see that a couple of lineages seem to have lost it again. And actually, we now know of probably 3 losses out of 9 lineages that we looked at. So this is a frequent thing. Nevertheless, we also know from previous work that, for example, in scuttleflies and hoverflies, the function of bicoid is basically identical to what I showed you in terms of for the midge, namely in preventing these symmetrical double abdomens. In Drosophila, it's not quite symmetrical because there is a second system, but that doesn't play a big role in these scuttle and hoverflies. So it's really a symmetrical phenotype, with the exception of the germline, which is maternally specified in all these flies and which is not duplicated. We now know of genes that prevent this phenotype from 3 species. So Drosophila is one example, and then these 2 midges that I show up there. And interestingly, these are completely unrelated molecules that prevent this double abdomen phenotype. And what I will do today is I will very briefly review some of the work on bicoid and some recent unpublished work also, mostly from the Thornton lab done, which was done in collaboration with several other labs, including ours. Then I will review the 2 other cases there before I conclude with what we can actually learn from this. So I start with bicoid. Bicoid, as you know, came out of the maternally effect screen that was designed by Erik Wischhaus and Nüsslein-Vollert. And because it was on chromosome 3, it was published by the Nüsslein-Vollert lab. And but the cloning actually was serendipitously achieved in Markus Noll lab, because bicoid has a little motif that hybridized to a probe of an unrelated gene, and so by chance they picked it up. And then there were, of course, the very famous papers by Driva and Nüsslein-Vollert, which seemed to firmly establish bicoid is a morphogen. And it was the first, and of course these are very, very famous papers. I just want to mention them. That wasn't the end of it, and people moved on, is nicely illustrated by a quick look at PubMed. Recently, I just screened for how many papers quote bicoid in the title or in the abstract, and there is about 620 papers. So that is still a very active field of research, and I became a member of this community fairly late, but already as a student, and I became very much interested in the question of when the bicoid gene evolved and how the bicoid gene evolved. And because I was young, I also asked the why question, that is, why did bicoid evolve? And we still don't know much about the latter, but we have made some progress for the first 2. And I put in these 2 men here because they were kind of important to me in inspiring me to work on this subject early on. Klaus Sander, the late Klaus Sander, was my undergraduate advisor, and Herbert Jäckle, my postdoctoral mentor. Bicoid, a few things you need to know in case you don't. It is a HOX3 related gene. duplicated probably in the cycloraphan lineage. I won't show you much of these data. All this is published. But it is well consistent with recent genome analysis that we have done in a number of lineages. And we thought that this duplication happens in cycloraphan flies already in 2002. We had a paper on this because we found that the closest homologue, the Zen gene, changes in a way expression in cycloraphan flies. While it is maternal and zygotic in many lower dipterans, it becomes strictly zygotic in higher flies. So the idea was that there is some sort of sub-functionalization, not functionalization in the sense that the maternal gene expression here would do anything of the ubiquitic work, but at least with regard to expression. And we also were aware that the binding properties, the DNA binding properties of ubiquitin are quite different from that of its sibling gene, and that goes back to work from Steve Haynes, who already, I think in 1989, proposed that the lysine in position 50 of the homodomain changes quite dramatically the ubiquitin DNA binding affinity. And very recently, this was re-examined by Joe Thornton in collaboration with Steve Small and Martha Bulick, and also a little bit of our input, where they try to ancestrally reconstruct at least the homodomain. You can't do it for the full protein, but for the homodomain, this is actually possible to some degree at least. And they systematically try to change the positions of let me see, changes that happened here on this branch. And but basically what came out in a lot of very nice biochemical work, but essentially confirmed Steve Haynes' idea that even in this ancestral context, it is mostly one amino acid change that makes a big effect. And you see that more dramatically here in this protein binding array from, oh, I'm sorry, oh, like this. Which shows you, so these protein binding arrays is basically you offer as beta all possible combinations of an 8-mer on a slide, and then you either take the ancestral homodomain of the last ancestor of Zen and ubiquitin, or you take the ancestral ubiquitin domain, so these would be the two ends of this long branch, and you change only one amino acid, and so if you take the ancestral ubiquitin sequence and flip back the lysine to this amino acid here, you basically obtain a very similar affinity to the Zen homodomain, and you can do the inverse experiment, and you see that too. So it's quite a dramatic change, and this is very puzzling because imagine this happens to an anterior, to a gene. How does it achieve a big important role in global axis specification? And the idea that came up early on was maybe it replaces a molecule like orthodentica, which also has a lysine there and which is a conserved head gene. There are problems with this idea because orthodentical function is actually pretty well conserved and much narrower than the function of these long-range morphogens. And so I was always a little hesitant to buy into this theory. I leave it there for the now, but I will come back to this. And so basically, just in a nutshell, ubiquitin is a new gene in higher flies. It encodes a homodomain protein, activates anterior patterning, no functions, other functions are known, and the DNA affinity of the ubiquitin homodomain shifted predominantly due to a single amino acid. I move on and tell you a little bit about these other models that we have been developing over many years. And so here we finally found a good approach that allows us to identify biquid-like genes in basically with very few prerequisites in other species, non-model organisms. And the approach consists essentially of cutting eggs and doing expression profiling of anterior and posterior halves. So we first did this for this chironomic niche that you see here on the left, which unfortunately has very, very small eggs, but you still can cut them and you can pull them and with some luck. So there were reasons to be skeptical that this would be clean enough, but it turned out to be actually super clean. And you see here in this volcano plot genes that are enriched in the anterior embryo on the left and genes that are enriched on the posterior on the right. And if you make an in situ hybridization, you can confirm this. And so I will talk more about this little gene that we named Panish because it is the gene that prevent these double abdomens, as we later found. So the gene expression is very, very similar to, at the transcript level here, to Drosophila bicoid. Initially tightly localized, and this transcript spreads out a little. We don't know how far, but maybe quite far. And then it disappears at mid-blastular transition, essentially, and is, to our knowledge, not active at any later stage. And if you knock it down, you get these wonderful double abdomens. You get them very efficiently. You can rescue that phenotype also if you inject messenger RNA of Panish. So we're quite confident we have the right gene, although the locus of this gene is actually a little more complex. So it is a gene that shares the first two exons with a nucleoside kinase gene, which happens to be maternally expressed, but as we know, has no role in axis specification at all. So this little Panish gene has a cysteine clamp, so it's not the homodomain protein. And there are only two cysteine clamp genes in the Drosophila genome. One is pangolin, so the TCF homolog, so the effector of Wnt signaling. And the other is GLUT. It comes with many, many names. But in vertebrates, both are, of course, much bigger families, but Panish falls in this pangolin TCF family. And it looks like this. It looks much smaller. It doesn't have a beta-catenin binding domain. It doesn't have an HMG domain. It only has this little cysteine clamp domain. And otherwise no obvious homology to anything. But the DNA binding affinity of that cysteine clamp is similar to that of the cysteine clamp of pangolin, and that's shown here again with just a summary of PBM, so protein binding microarray data. So essentially, these genes seem to recognize similar things, although the pangolin gene clearly becomes activated after Panish has done key jobs, as I will describe now in a second. So what are key jobs of Panish? Well, it represses posterior determinants, such as talus and caudal. And so if you knock it out or down, you duplicate these posterior genes. And what is very interesting to us in this context was that the zygotic talus gene, which has here a posterior gradient, actually posterior to anterior gradient, you see it up in the left there, is an expression that we do not find in species with bicoid. This is something that we see only in lower dipterans. And if you knock down this zygotic gradient, you actually get a double head, and that's what you see in the lower panels. So that led us to, with other data, to a model in which localized Panish activity represses the activation of posterior genes, probably under the control of ubiquitous activators. and that maybe Taylor's is some sort of a toggle here in the polarity. There's one additional thing I want to mention, and that is, again, we are looking into a gene that is very newly evolved, very recent. We only find it in a subfamily of chironomid midges, which is quite intriguing because in much of the earlier work done by Kalthoff, they used a model outside where we find our punished gene. So we still may not have found the gene they were hunting at the time, although we have a better idea what this might be now. So the summary of this is, so we have a new gene again, evolves in higher chironomids, if you like, and it codes a unique cysteine clamp protein. It works as a repressor, probably, so in a permissive role because of the phenotype that we see in Taylor's knockdown, which gives you a head in the posterior end without punish. And we don't know any other function. So if you delay the knockdown until the midway through blastoderm stage, you don't see any effects anymore, at least through embryogenesis. And the DNA affinity is similar to its closest homologue, TCF. That was my second chapter. Now I will talk about the third case before I come back to some of the earlier questions. So here, this is the model. It's Clockmere, albipunctata, and I put here up the life cycle because I think actually I have looked at many different species in dipterans, and we have been lacking for many years a suitable model for doing developmental genetic work in lower dipterans. And I think this one has the potential of becoming a genetic model system for the lower dipterans, for those who are interested, for example, in the evolution of developmental mechanism. And the reason why I'm saying this is predominantly because you can mate these guys, single pair matings, and you can keep the stocks in petri dishes. And that is basically a huge relief if you compare chironomers, like mosquitoes, they all have, you need to keep the larvae in aquaria, it's much more elaborate. So this is a low budget system, very nice. And the genome makes big progress. We will hopefully soon have a high-C assembly too, and the transcriptomes available. And you can, that big limit was to inject the guys early, which is a key technology, obviously, for developing non-model species. And that finally has been solved too, so we can knock down genes and we can overexpress genes. And here are the data. So same approach. And look at this volcano plot. There's, again, one single gene localized highly, and it's odd paired. That's actually a gene that is known as a perul gene, and segmentation gene, and it is related to the Zik genes of vertebrates. And there's a couple of other things that are noteworthy, and mostly, if you look on the posterior end, there's hardly anything localized there. So these guys seem to have lost their maternal germplasm, and I'll show you evidence for this in a minute too. So, so odd paired is interesting because it comes in two isoforms. And there's a maternal isoform, which is shown here on the top, and then there is a zygotic isoform. And these transcripts, the open reading frames, are almost completely identical. If you look at the only difference is that there's a tiny bit missing at the N-terminal end of the encoded protein, but otherwise they are the same. And the expression seems to be, for the maternal transcript, seems to be strictly localized, and that's the only thing we see. The transcript doesn't seem to be used apart from this context. And then if you look at the zygotic transcript, so these are in situ hybridizations that are transcript specific. And when you look at the zygotic one on the lower side, you see that it has this broad domain, which is canonical for the dipterans at least. Odd pair has a special role in perul patterning. It actually makes a transition from perul to segmental expression at the beginning of gastrulation. There's a couple of recent papers on the topic by Eric Clark and Michael Eckham. So here's the phenotype. You knock it down, you get nice double abdomens, and you see the second row up there shows you a nanos expression, and you see that the germline duplicates too. So that's pretty strong evidence that we have an inductive system for germline specification, zygotic. And of course, you see the cuticle perhaps below there. That is all, seems pretty low tech, but it is actually quite a lot of work to develop these protocols in a way that you can see things in a nice way. And the zygotic function, so this on the right was only the maternal transcript knockdown. If you knock down the zygotic transcript, you get a conserved function in pair rule patterning, which seems very similar to what we know from Drosophila. So when you take that gene and express this on the posterior end, you get a very nice double head. And this is the living embryo. You see it's on both ends. You see the pulsating pharynx. And so you can see that here with molecular markers. This shows your orthodentical expression on top and cuticle on the right. And then you can start to ask more questions. You can ask, now you have a nice essay. Once you can do this, you can, of course, start to clip off things and test whether a modified gene can still induce a double head. And that is true for a lot of the truncations on the N-terminal end. So it's quite robust to that. It is much more sensitive to truncating the C-terminal end. And also, and that is important, when you delay the knockdown of the maternal if you inject after the very early stages, the genes can no longer induce head function. So what it means is that this function or this capacity to induce head development is pretty narrowly defined in a time window very early in development. And so we have also looked a little bit into where it is conserved, and I only mention it seems to be broadly conserved in moth flies, including sandflies, which are an important vector of human disease, leishmanias. And but I bring this case up not only to show you that it is conserved in moth flies, but also because we also find two transcripts there, and in this case, the open reading frame is identical. So we think actually this little truncation that we saw in Clogmere might be just some secondary modification. And the next experiment, I think, nails down it's a simple experiment, but it nails down a very important point. We tested now odd-paired homologs. We took the zygotic version of Clogmere, we took the one from the sandfly, we took the one from Chironomus, we took the one from Drosophila, and they all can induce double heads. And there's, of course, some negative controls. You have to make some precautions. You have to use the endogenous Cossack sequence of Clogmere to get efficient expression, but otherwise you can induce these heads, and quite nicely, they'll just look the same. And so what this tells us is that this function was acquired by adopting a new isoform, localizing it. Protein change did not matter, right? So that's, I think, quite important. So we have a maternal isoform that adopted control of embryo polarity, while the zygotic isoform retained ancestral function in segmentation. And we also have evidence that this evolved via alternative transcription. No protein change was required. So that brings me to general conclusions of what I think we can think about here. And one important one, I think, is that genes, even a gene like bicoid, can be younger than the function that it represents. So it is not excluded at all that a bicoid inherited its function from a localized Zen transcript. Indeed, it would have had 20 million years to evolve that, because that is the stem of the cycloraphens that we can't test with extant species. So we may indeed overestimate the role of protein divergence as primary drivers for entirely new gene functions. I might put this out here as a hypothesis, but I think it is a very strong case for, in fact, co-option, in this case through alternative transcription. And that might be also interesting for the simple reason that alternative transcription, particularly in vertebrates, is extremely common, apparently. So recent genomic studies find this in more than, or about 80% of human genes. So it's a very common mechanism. And so let me come up with a couple of open questions here. So, of course, we don't really know yet what drives the replacement of anterior determinants. Apparently, that happens very often, and it involves somehow this very tight localization. So, the idea would be that as a transcript becomes more and more localized, it must be some feedback enhancement. Can become more and more dominant in controlling anterior patterning until it kicks out what used to sit there. And these genes may actually be quite unrelated. And obviously, the function of these genes resides in how the target genes respond to it, not to the gene itself. There's also, of course, the function. So if you apply this to these questions to how did Panish evolve, or how did Biquid evolve, I think we need to look more broadly, probe more lineages, to look for obviously localized and functionally equivalent pangolin transcripts in the case of Panish. And I don't know if there is a chance to ever find a localized Zen transcript, because there are 20 million years for which we don't have data. But I think we can learn something about how Biquid evolved. If you look, for example, at the intron positions in Biquid, there are two conserved intron positions, and you can explain them very nicely if you take the zygotic Zen transcript that we find in lower flies, and duplicate that so that it captures the maternal promoter of the second transcript. Drosophila biquid has 3 introns, but others only 2. So I think we can learn more. We just have to look more deeply and more in detail. And this shows you a couple of projects that are ongoing. They're shown here in yellow, so I can hopefully tell you more in not too far future. And with that, I would like to acknowledge the people. I have put the names on the slides. If you don't seen them, so there are different projects. I would like to point out collaborators that have contributed. So Biquid work was done in Joe Thornton's lab in collaboration. We collaborated also with Martha Bulick and a couple of, and I would like to mention in particular José Ribeiro's lab, who gave us access to sandfly cultures. And I have a long appendix, but I think I stop here because we are all hungry.